So good morning everyone, I am Dr. Ashish Pargaonkar and I come from our applications lab which is called Center of Excellence in Bangalore, and I take care of mass spectrometry based proteomics, metabolomics, biopharmaceutical applications, taking mass spectrometry as an approach. So basically what is a protein and other things, you know everyone, like it's a chain of amino acids, multiple amino acids, polypeptides. They can call it as a peptides, polypeptides, and then proteins. So proteome denotes the protein complement expressed by a genome. And then proteomics is the study of, so what is the difference between this term and these terms? Proteomics, metabolomics, transcriptomics. When it becomes omics, why so omics? So these terms makes it omics. Whenever you measure the quantitative changes of a protein complement, it becomes protein. Of a metabolite complement, it becomes metabolomics. Of RNA, transcriptomics. Glycans, glycomics. Lipids, lipidomics. Omics. Anywhere where you measure the changes and you quantify them, it becomes omics. Okay, so that is how it is. So that's why. And then of course there are various proteins composed of 20 common amino acids in different combinations. Okay, and these amino acids are basically linked together by a bond called a amide bond, or also called as a peptide bond. Okay, so they link together in a peptide bond. So we take all this information is very critical because these are the basis how we interpret in mass spectrometry. Okay, so we must know what is actually happening. Okay, if we know that, we are very well understand what exactly is happening in the mass spectrometry and how to interpret that. All right, so this is critical. So what we do exactly there is, it's not that complicated, it's quite easy, but only thing like there are multiple steps. First step is to identify. You have proteins of two states, you first identify what are those proteins. We have only numbers right now, right? We have to get some identity, okay, this protein and all those things. Then you have to characterize whether the identity is correct or not and where it is relevant, and then you have to quantify, okay, how much of it, either relatively or absolute. Okay, so for protein identification typically is what we follow is the bottom up approach. I'll come to that. A peptide mass fingerprinting, you digest a protein with some protease, you get into form of peptides, small peptides, you trigger an MSMS of the small peptides, again fragment it. So you've broken down the protein into small pieces, collate it together and submit into, so bottom up. Okay, so you are taking everything together and then submitting it to database. So that is how we do it. And then you get the identity. You have started with some mass number, you have fragmented that with another group of mass numbers, you submit it to database and get some name, what exactly it is. Okay, that is what it is. The second characterization is you sequence it, you identify or characterize what are the modifications. The protein can be in native form, it can be modified forms. So, and what are the sites where it is modified. So that is what the characterization, and then interactions, and then quantification, either by doing a absolute using iTract sort of technology or label free, differential. So what is the analytical challenge? When we say proteomics we have to do, what is the challenge? And then how to address that challenge? So the challenge remains same what we discussed in the previous presentation also, like there are almost close to 30,000. If we say human, 30,000 expressed gene in human genome, and each cell type is estimated to express almost 10,000 genes. But not one gene is equivalent to one protein. One gene can express many proteins also. So due to protein processing modifications, estimated to be 20,000 to 30,000 proteins per cell type, and there are about close to 250 different cell types. So you can see the dynamics and the complexity. Slowly it is increasing. So that is the challenge. So you have to actually typically identify close to 30,000 proteins in one cell. So you can imagine there are 250. So it's quite complex. So how to address that now? With comparison to genomics, if we say, you cannot amplify proteins. Unlike your genes, DNA or RNA, you can amplify, but you cannot do it. So you need a high sensitivity analysis. The dynamic range, concentration. A protein can be having various concentration. There can be a low abundant protein, there can be a high abundant protein at the same place, same point of time. Okay, so you must have to address this aspect also while analysis. While following an analytical approach, you must address this aspect also. This is one of our criteria to address this. This is one of the biggest challenge. The protein modified and processed. So detect and identify multiple species of the same protein. Same protein, various species, different isomers, different modifications, all those things, okay. And differential solubility. So generality of analysis. You cannot generalize for this type of tissue samples. This is the method. It may not be true for other type of analysis, okay. So that is how it is complex. So to overcome that, we follow the LC-MS approach, which reasonably addresses most of the challenges, okay. Basically, because it can be first automated. You can do multiple runs and you can automate it. You need not to sit there and do it. You run overnight and morning at least you can compare results and see like what is happening. Allows in-depth analysis. It is highly sensitive. It has wide dynamic range, okay. And you can run multiple proteins in one sample at a time. And it is in a way compatible to database searches, like data as such can be submitted into database and you can get the identity annotations. Okay, so there are different strategies of MS-based proteomics. As we talked, this bottom up, you digest. This is most commonly used in electrospray ionization type of approach, where you have the protein or mixture of proteins, you digest it, fragment it, do an MS-MS, hybrid type of mass analyzers. Okay, when we say hybrid means quadrupole, time of flight, let us say, coupled with a collision cell, where you can do an MS-MS and get the peptide masses, accurate mass, and then you identify the protein, okay. The other one is top down, where you fragment protein at the intact level itself and get the fragments at intact level itself and then try to identify. That is another approach. Most popular platforms are either TOF or tandem TOFs. When I say tandem means quadrupole coupled with the time of flight, so quadrupole time of flight, tandem. Then are ion traps, where you have MS-MSN capability, hybrids, okay, and then magnetic sectors. So there are various types of popular platforms, but I'll just concentrate on something which we have in Agilent portfolio, which is being used for proteomics, which is a quadrupole time of flight, Q-TOF type of instruments. Why we use Q-TOF, even though we have a triple quad also, but we still say like for proteomics we need a time of flight type of instrument. Why so? Because of the three reasons why we need a time of flight. One is sensitivity. As we understood, like there is a challenge that we need to have a sensitivity. Then there are two things which is very critical in the time of flight, which probably a quadrupole type of analyzers might miss, is the mass accuracy and resolution. So typically, like in a time of flight, quadrupole time of flight instrument, you have a quadrupole and then a time of flight where the ions will travel like this and the time is measured, coupled with a collision cell, where you trigger an MS-MS. When we say, what is the word we typically use? LC-MS-MS. Okay, so there is a LC component, liquid chromatography component, also coupled to an MS. Okay, why so? Because we want to separate. What exactly liquid chromatography does? It has a stationary phase, reverse phase typically, and you separate peptides based on their chemistry, hydrophobicity in this case, or different chemistries for different chromatography. Okay, so you separate your peptides. The outlet of an LC is what? Liquid. But we are saying here that my mass spec cannot see mass as such. It has to be charged, meaning the language which my LC is talking cannot be understood by my mass, isn't it? So we need an interface which is called as ion source, which we just now see, or in this case electrospray ionization. That is an interface where the liquid is evaporated and only charge analyte, in this case peptides, gets into the into the mass spec which is under high vacuum. Okay, and it travels through the quadrupole, then you can trigger an MS-MS and then it can be measured in the time of flight. Okay, so why quadrupole and why time of flight, maybe we will discuss when we actually go into the lab. Exactly when you can see it, like there are various modes, and then you can realize, okay, why not only TOF? If TOF can do everything, as I told, okay, it's accurate mass resolution, then why we need a quadrupole? Okay, so we will see that. This is the instrument, something like what we have seen in schematics, a quadruple time of flight type of instrument. Okay, where you have quadruple, as I shown you in the presentation, then you have collision cell, okay, and then you have a time of flight. Okay, so quadruple and time of flight are in tandem. Okay, meaning in tandem with respect to doing an MSMS. Okay. So there are two different analyzers, quadruple and time of flight. Okay. And that is coupled to an HPLC. That's why we call it as LC-MS. HPLC-MS. So you know there are versions of proteins in the samples. Okay. And if you follow a bottom-up approach where you digest the protein with an enzyme, okay, and then you do an MSMS, all those things, and you then try to search it. So if I digested the protein and thousands of proteins becomes 10,000 of peptides, okay, and I just directly subject into MS. MS is running some, it is scanning, right? It will have some limitation. It can see up to this, not beyond it, something like that, just to make you assume. Okay, it will have some limitation. So 10,000 peptides coming at a time, it might not see. Okay. So it might see some of them, it might miss most of them. If at all it is coming like that, in a fraction of a second. To reduce that complexity, we use a separation technique before an MS so that your peptides get retained based on their hydrophobicity in the stationary phase, which is we call it as HPLC column. If anyone has not used an HPLC, it looks like this. It has some stationary phase, typically silica-based CAT, depend upon what type of chromatography you are doing or what type of chemistry you are running. Okay, so it gets retained based on their hydrophobicity, and then you elute it or you remove those peptides at different time intervals. So 10,000 peptides, 2,000 will come at 1 minute, next 500 will come at 2 minutes, like that. Then you split all those things, and then your TOF can see them, or your MS. Let us not say your MS can see them with its full capacity, and you won't miss it. That is one. You reduce the complexity of it. So as an HPLC, if you see configuration, HPLC basis is it's a separating device. Okay. There is a module called as pump, binary pump typically two pumps. What the purpose of two pumps is? To pump the mobile phase. Okay. And typically what type of mobile phase we use? In the case of peptides, it is hydrophobic interaction. Okay. And based on hydrophobicity, it will bind to the stationary phase, and then you elute by changing the polarity. Okay. We have pump, then we have a column compartment where you put the column. Typically, it's heated. You can heat it to have better separation. Then you have an auto sampler. If you recollect my slide where I said it can be automated. Okay, so this can be automated. You can put hundreds of samples in that. You define your runs, and you can carry out your other works. And by the time your analysis gets over, automated. Okay, but meaning is what I mean to say here is like if you put 100 samples and if your run time or the separation time taken to separate is 30 minutes, let us say. By the time the number of 100 sample number comes, it is 300 minutes, isn't it? Or more, it is more than that. Okay, what it means, samples might degrade by the time its number comes. Okay, so to maintain that into constant temperature, you have a thermostat. So you maintain your temperature of the samples in the autosampler at 4 degrees Celsius, so that your sample doesn't degrade during the process of analysis, if you are actually running the automated type of analysis. And then there is the DAD, that means UV analysis. Okay, so typically there are two pumps, as I told you, in the pump configuration. Pump A consists of water with formic acid. Okay, and pump B consists of acetonitrile, typically with 5+ formic acid. Okay, so why formic acid? To give protons. Okay, why so? Because I know my analytes are peptides, which are having many amide groups, okay, which can take protons and gets charged. And I know the basis of mass take is what? I need charge, mass to charge. Unless my analyte is ionized, this is blind. It cannot see. Okay, so the prerequisite of an MS analysis is that your analyte must get charged. Okay, and in electrospray typically your analytes gets ionized in solution, meaning in the solution itself it gets protonated, and ESI is generally facilitated the ionization. It allows, it creates an environment where your ions moves into MS suitably. Broadly, I would say. The pumps will pump the mobile phase, which is A is water, let us say. So, by loading the sample, we'll use more of aqueous. Okay, so that your peptides get retained on the column, stationary phase. If this is my stationary phase, depending upon hydrophobicity, 10,000 peptides, few will bind here, few will bind there, and they'll get spread across the column. Okay, based on their hydrophobicity, and they'll get retained. And you can run for a while with aqueous only, so that any salts or other things get washed off, and only you get enrichment of your peptides. Then what you do is slowly increase the acetonitrile content, which is also called as organic step gradient or organic gradient. You increase slowly the organic content and reduce the aqueous content, so that the polarity is now changing, and depending upon their binding capacities, they will cleave off from this and then start selecting. Okay. If this is so, why we can it means I can measure the peptides at UV itself. Why we need MS? Okay, anyway it is retaining, everything it is doing. It is retaining also, eluting also, and you can measure it in UV also. Then why need the MS? Anyone has used HPLC? So if you have used it, what is the readout of an HPLC? If you inject the sample, how you see the chromatogram? Means I'll ask you a question. If I give you a chromatogram of an HPLC UV, if there are 6 peaks, what it means? 6 compounds. You get 2 dimensions in your analysis. One is compound, another one is retention time. Okay, based on compound you can do that. But will it serve my purpose? Because in peptides, in unknown scenario, I do not know how many proteins, okay, and how many peptides, and different same peptide, different peptides from different proteins can correlate with each other. That is also possible. Okay. So I need another dimension to that, to see that, to look beyond the field. What is that? So for that, the third dimension which is given to that analysis is the mass spec, which is the mass to charge or mass. Okay. So when the one peak of HPLC goes into the mass spec, it sees beyond that 2 dimension, which is mass, and it will tell you that in one peak is not one compound, there are 100 more masses. Okay, within that peak. So as I told you, like it runs through the column and then it dilutes in the form of liquid like this. Okay, it is coming like this. So now right now the method is like it is running at 0.4 ml per minute. The flow rate is 0.4 ml per minute. You can go to 1 ml per minute. But of course, with this source only, I am not going into details of that specification of the source. This source particularly, but the other sources, MS in general, you may not be able to go beyond 0.5. So right now, at initial, when I have to load sample, there is a flow which is going on, which is 3% of organic and 97% of aqueous right now. Okay, so more of water right now because I have to inject the sample. So it goes into, so as you see, what is the output here? It's the stream of liquid. But what it measures? Mass, ions. So the language which is talking is not understood by this right now. Okay, so we need some sort of device which converts this liquid into ions. So that is comes the role of an interface, which is also called as ion source or atmospheric pressure ionization technique, and one type of atmospheric pressure ionization is electrospray, which is this. Electrospray ionization, or also properly called as pneumatically assisted electrospray. Why so? It is something called as spray. So the stream of liquid is going into the needle, and then it is sprayed. Why so? Because our analytes already we know it's in the charge. So the charge particles or charge analytes are already there in the liquids. So I want only those parts to get in, not the acetonitrile, formic acid and all those things. I want to get rid of that, and the ions should go in. That is what the objective is. So what is being done here is a stream of liquid, then there is a nebulization gas which is nitrogen, then that liquid is sprayed like Bivon spray, sort of, something like that. So you are pushing some gas around that nozzle so that it gets sprayed off. So you have split the flow, split the stream of flow into small spray, and the source is heated at typically 300 degrees centigrade. Okay. So when the spray is happening, imagine a droplet of liquid with different charge peptides inside that. It has came out into that high temperature. Acetonite will get evaporated slowly. The positive charges came closer. The moment it gets closer, they will repel, they will explode. Coulombic explosion we call it. That is the phenomenon in electrochemical. You get a Coulombic explosion. And many such explosions are happening in fraction of seconds. All are gone. And then all gets evaporated, dissolved because of high temperature in that. Yeah. Okay. Nitrogen. It has to be inert gas, otherwise it should not react and form something else inside. Right? This is how the spray will happen. Yeah. This is you see spray. I showed you the droplet. Now you see the spray. You could able to see. You see, this is how it happens. Now one drop has become like this spray, and inside there is high temperature, 300. You can imagine how fast, unimaginable speed it has to. So there are different analyzers. Quadrupole is one type of analyzer. Okay. Analyzer of mass. Mass analyzer. So quadrupole is also called as mass filter. Mass filter. It's also called as mass filter analyzer because of the way it behaves. Quadrupole means what? 4 rods. Quadrupole. So 4 rods like this. There are 4 rods like this, positioned like this, at some length. Okay. So what is the logic? It is also called as mass filter. Why? Because it has the ability to filter mass. If you say look for mass of 500, it will filter everything away and only filter 500 or only allow 500 to get inside the quadrupole and travel through that. Travel through 4 rods. How it does that? These 4 rods are at different electronic states like RF and DC voltage. For example, this rod and this rod, alternate rods are in RF mode. The moment I say look for 500, okay. As a user, I will say only 500. But for electronics, it understands how much energy, electrical energy it needs to attract mass of 500. So it will apply that. The ion will come here. 500 ion will come here along with 10,000 other ions. Okay, but it has applied efficient energy to capture only 500. So what will happen? Out of 10,000, maybe 9,000 gets lost. They will hit somewhere and get lost. Along with 500, more 9,000 ions will get in somehow because of the voltage. So moment it comes close, the polarity switches. This becomes DC, this becomes RF. So now this, instead of attracting, it will repel 500, and this will attract. So it will travel like this. The moment it's like during this, probably the other 5,000 will get hit here and then get lost. Then again polarity switches. Then it comes here. So like that, it will try to travel in the quadrupole and selectively, and other 10,000 will hit somewhere at the other and get lost. So in this way, you can filter mass with very specificity. Let us understand in general first functionality of why we use this and then applicability. And these are very specific questions because you have experience, that's why you are asking. Some experience I could sense that, but most of you may not have experience. But at least you should know what exactly is quadrupole doing. So quadrupole is a filter. Okay, it's a mass filter. It's very specific because of that thing. It is selective, hence sensitive. Because that's why it is used for targeted analysis. You have heard the name called triple quad. Quadrupole, collision cell, another quadrupole. Instead of time of flight, if you have put another quadrupole, it becomes triple quadrupole. And when you say two mass analyzers with the functionality of having an MS in the terms of collision cell, you call them as in tandem MS, because they are talking to each other. They are in tandem with MS in between. if there are two different analyzers, like here in this case quarter pole and time of flight, then it is also called as hybrid mass analyzer, because quarter pole in itself is a mass analyzer, time of flight in itself is a mass analyzer. Two different mass analyzers coupled with a functionality of MS, it becomes hybrid mass analyzer or hybrid tandem MS. Because in this case, quarter pole and time of flight are also talking to each other with MSS, also called as data dependent acquisition and all those things you might have heard them. And we need to have two different analyzers. Okay. For our case, in unknown scenario, we know that there are different types of protein, which are, and I don't know about that protein. So first place, I want to establish the identity of that protein. So I need accurate mass information. I cannot use a triple quad type of device which has unit mass resolution. It cannot see beyond 1 Dalton difference. So hence it is not accurate. So if I submit triple quad data to a database, there will be many peptides of the same mass but different sequence. It will pull out all the information because it doesn't have accuracy. It will see as 500 and 501 only, and you will get redundant information. For the same mass, you will get hundreds of peptides. You started with nowhere, you end up nowhere. So what to do next? I don't know which out of this 50 peptides what is correct. But the moment you put time of flight, 500 becomes 500.12345, and the fragment of 500.12345 becomes 10 different fragments of accurate mass. So it is absolutely unlikely that a peptide of that sequence is giving some different MS pattern with different masses. So the match will be absolutely perfect. You started with nowhere, you ended with somewhere which is reliable. That's why we need time of flight. Okay. So as a functional, now as an applicability, how we apply that? So broadly, the Q-TOF can be run in 3 different modes. Okay, modes of acquisition, if I say so, in terms of application. First is TOF only mode, TOF only. So as an applicability, we can run in 3 different modes. First is the TOF only or MS only mode. When I say MS only means my quadrupole collision cell is not behaving as they have to. Meaning quadrupole will not filter and collision cell will not fry. So all ions which are going in, they will simply pass through the quadrupole, simply pass through the collision cell and go to the TOF, and TOF will scan. PMF type of approach, peptide mass fingerprinting, based on only mass. You got the accurate masses of all the peptides, then you search. That is one way. Nowadays, mostly no one is using that PMF approach because of the same simple reason that again redundancy will rise. Okay, based on only accurate mass might not be sufficient. You may have to trigger anything. Then comes the other approach which is called as data dependent. Data dependent approach. There we take advantage of both the analyzers, quadrupole as well as time of flight. I have some sample which is complex. I don't know how many peptides, which peptide is coming at what time, at what concentrations. I have no clue. I cannot guide my quadrupole to select what and when, what time. So no clue. So let instrument decide. Depending upon the data, as it comes, real time, decide to fragment. The decision engine of the time of flight will decide what to fragment at what time, depending upon the inputs given by the user. So user will define that look for peptide isotopic pattern. Look for only those peptides with some certain abundance level threshold. Okay. So based on that, it will look for that. In MS only mode, let us say this machine has the capacity to scan from 5 M by Z to 3200 M by Z. Let us say 3200 M by Z. That is the range of a machine. But I know my peptides, which is doubly charged, cannot go beyond 1200. Maximum 1200. Okay. So if I scan, if I'm scanning, my TOF is scanning 1 spectrum per second, meaning my cycle time is 1 second, means it will start from 5, it will come to 3200 and come back. It happens in 1 second. It will take 1 second time to scan complete whatever I define. And I know my peptide will not come beyond 1200, meaning I am wasting some time in scanning something which is useless. Instead of that, I can spend the same time to capture something which is informative. So I take advantage of the protocol there to say, look for mass from 200 to 1200 only. So it will filter others and it will only allow 100 to 1200, and that 1 second I will apply till 1200 and come back. So that is the scan time. In data dependent acquisition, this same scan time is split into two different experiments. Experiment 1 is scan first, let everything go into the top, and in this scan, pick up the precursors based on the decision engine, based on the depending upon the data which is coming. Meaning threshold, charge states, okay, all those filters in place. If it qualifies, let us say 10,000 ions are scanned. Out of that 10,000, only 8 or 10 is relevant, is peptidic. It will pick up those and it will tell, the TOF will tell to the protocol in the same cycle, filter only those 8 now. Okay, that is why it is in tandem, it is talking. It has scanned, it has told that pick 8, by coming back, it will pick up only those 8 in the same cycle, fragment those and give it back. So now, the TOF will stitch the information because it has the scanning, it has the MS of 8. So it knows that which are the precursors at MS level, and it knows what are the fragments of that precursor. So it will stitch the data, so it will have an MS and MS of that peptide stitched together, and you can, as a user, I can see it comfortably on my desktop. The third one is data directed or targeted MS, which is possible because we have a protocol time of flight. When I say targeted means you have to define what are the masses you have to select, it will trigger MS and give it. What it means? It means a quadruple time of flight device can also be used for quantification. Okay, of course, it cannot be that sensitive as a triple particle because of the same reason that the third analyzer is scanning. It is not a mass filter like triple particle. Since it is scanning, it is wasting time. It is scanning something which is not relevant, and hence you may lose something. That's why it is less sensitive. It is not that sensitive as a quadruple.